Behavioral Neuroscience, Chapter 12, Segment 3. Androgen insensitivity syndrome, or AIS, is a syndrome caused by a mutation of the androgen receptor gene that renders tissues insensitive to androgenic hormones like testosterone. Affected XY individuals are phenotypic females, but they have internal testes and regressed genital structures. Women with AIS develop breasts during puberty due to a high estrogen to androgen ratio. However, they lack ovaries and a uterus, do not menstruate, and are infertile with a shallow vagina. Babies are occasionally born with a rare genetic mutation that disables the enzyme 5-alpha reductase that converts testosterone to DHT. An XY individual with this condition will develop testes and normal internal male reproductive structures because testosterone and AMH function normally, but the external genitalia will not masculinize fully. Known as guevedosis, a nickname for individuals who are raised as girls, but at puberty change appearance and begin behaving as boys do. The only behavior exclusive to one sex is childbirth in women. There is evidence that the same hormones that masculinize the genitalia of male animals in utero also masculinize the brain to affect later behavior. Female guinea pigs exposed to testosterone in utero did not show lordosis in adulthood. The organizational effect is a permanent alteration of the nervous system and thus permanent change in behavior resulting from the action of a steroid hormone on an animal early in its development. A sensitive period is the period during development in which an organism can be permanently altered by a particular experience or treatment. In mammals, puberty can be viewed as a second sensitive period. The organizational hypothesis says that a single steroid signal, androgen, diffuses through all tissues, masculinizing the body, the brain, and behavior. Only animals exposed to androgen both in development and in adulthood show fully masculine behavior. In a chemical reaction called aromatization, the enzyme aromatase converts testosterone to estradiol. The aromatization hypothesis is the hypothesis that testicular androgens enter the brain and are converted there into estrogens to masculinize the developing nervous system in some rodents. An alpha-fetoprotein is a protein found in the plasma of fetuses in rodents. In rodents, alpha-fetoprotein binds estrogens and prevents them from entering the brain. Hormonal masculinization must be accompanied through androgen receptors rather than estrogen receptors. A hermaphrodite is an individual possessing the reproductive organs of both sexes, either simultaneously or at different points in time. Hyena fetuses are exposed to high concentrations of androgens. Females are aggressive, and mating tends to be a tense affair. Sex differences in the number of synapses were identified in the preoptic area of the hypothalamus. Sexual dimorphism is the condition in which males and females show pronounced sex differences in appearance. The syrinx is the vocal organ in birds. It is controlled by the RA nucleus. The RA is controlled by the HVC, or higher vocal center. Male canaries and zebra finches use elaborate songs to attract mates, but females do not. This is affected by androgens. In canaries, the HVC and RA shrink in the winter as testosterone levels decline. The sexually dimorphic nucleus of the preoptic area, or SDN-POA, is a region of the preoptic area that is 5 to 6 times larger in volume in male rats than in females. There is growing evidence that androgens exert epigenetic effects. The spinal nucleus of the bulbocavernosus, or SNB, is a group of motor neurons in the spinal cord of rats that innervate striated muscles controlling the penis. The BC muscles of female rats die before birth. This is also the case with castration in males.